Um, it's a real pleasure and honor to introduce Chris. Chris started off at Southeast Missouri, a Red Hawk, as I learned, and we have turned him into a Badger. So he came to graduate school and was in Sean Carroll's lab, and then did a postdoc with Mark Johnson, who I think was at WashU and then moved kind of in the middle to CU in Colorado. And then the department hired him back in 2011. So Chris quickly rose through the ranks. He was a star in the making. He's a star now because that was then and this is now. So he's got lots of awards, lots of papers, lots of talks. So I don't want to summarize anything, but you can see just off the titles under his name that it's got a lot of stuff going on. So I'll just sit down and hand it over to Chris.
Well, thank you very much, Jerry, for that introduction. I want to thank everybody for coming. It's a real pleasure to be able to address this audience. This is the first time I've given a laboratory of genetics colloquium since graduate school, where my job or promotion has not been on the line. So I'm very pleased to be able to talk science to you with there being virtually zero chance of me getting fired today. Oop, I'm doing something wrong. There we go. Okay. So the first thing I need to tell you is that yeasts are diverse. And so we look at the entire subphylum, Saccharomycotina, which contains the model yeast Saccharomyces cerevisiae up in the upper left-hand corner there. But it actually contains as much genetic diversity as the entire animal kingdom, and actually quite a bit more than the plant kingdom, right? So if we look at amino acid substitutions along the x-axis here, we march out the yeast, and we pretty much capture the entire animal kingdom, nearly the entire plant kingdom. Now, we've learned a lot about genetics from Saccharomyces cerevisiae, the model yeast. It's taught us a ton, but what can other yeasts teach us? So we're going to focus on carbon metabolism because yeasts are one-celled fungi whose carbon metabolisms vary widely. And so energy harvest and storage is really one of the most fundamental challenges that organisms face. In particular, it's really crucial for heterotrophs like fungi and animals because they're lazy. They have to take advantage of whatever energy has been stored by plants and whatever carbon sources they're providing. And so generally, they only consume a subset of possible carbon sources, and they're actually choosy about which ones they prioritize. They also make different decisions about how to metabolize, what to do with them. Sometimes some yeasts will completely respire carbon sources all the way to carbon dioxide, whereas other yeasts will ferment or perhaps even make other modified compound storage molecules to various degrees. So really both sides of this coin are essential to yeast carbon metabolism, capabilities on the consumption of sources and the modifications to carbon products. And of course, ultimately, all of this is encoded genetically. We also exploit these carbon sources and products in various industries for delicious beverages, such as beer, and also for sustainable biofuels and bioproducts. And at the end, I'll tell you a couple of stories about traits relevant for there, which I've color-coded here. So a few years ago, we obtained a grant from the National Science Foundation to fund the Y1000 Plus project. And this was an ambitious project to really create a genotype-phenotype map across more than 400 million years of evolution. And so the workflow went like this. We took advantage of taxonomic practice, which is that when you describe a new species formally in the taxonomic literature, you're required to deposit it in publicly accessible strain banks in 2 separate countries. And so we were fortunate to collaborate with Cleet Kurtzman down at the USDA facility at NRL. Unfortunately, he has not survived the project, but we still have really great relations with the folks at NRL. We took all of those taxonomic type strains deposited by taxonomists over the years, brought them up to UW-Madison, up I-39, and then we sequenced their genomes using the Illumina platform. And of course, yeast genomes are pretty small and snap together reasonably well. We also conducted high throughput phenotyping using a plate reader and stacker that can do nearly 5,000 growth curves simultaneously. And so this is getting us quantitative information about what yeast's capabilities are. And then, of course, we end up with a big data matrix. And this was probably the vaguest part of the grant, is how we were actually going to do the magic once we had it. But hopefully you'll get a feeling of it by the end of the talk today. So as the genomes were starting to roll in, my postdoc, Dana Opulente, who's really trained more as an ecologist, she really wondered if she could do things with the trait data alone. And so we asked a simple question, which is, do yeast traits covary? And what is framing our thinking here is this concept of trait syndromes from ecology. And so I'm showing here one of the most famous trait syndromes is domestication syndrome. But there are others. The Pacer Lab studies island syndrome. There's a plant resistance syndrome. And the idea is that there are these suites of traits that go along together ecologically. And so when we domesticated dogs from wolves, they became docile, got floppy ears and big doe eyes. And the same thing happened over a very short number of generations when Belyaev did this in Russia with the Arctic fox. And so there's this idea that there are these traits that if you impose selection in a similar ecological environment, you get a similar outcome. Now, I want to stress that for yeasts here, we're not talking about domestication, even though there's been a lot of focus on, of course, bioproducts and fermented beverages and domestication. We're talking about things that happened because of 400 million years of evolution in yeast ecology in nature. So to test this hypothesis, we again took advantage of taxonomic practice. And so we got the traits from this book written by a couple taxonomists, including Cleet Kurtzman, and we recoded them as ones and zeros. And so the initial trait matrix was 784 species, and then we have rows with 48 traits. Now, this is the data that actually underlies all taxonomic descriptions. Now, they thought that they were basically coming up with a key to key out and identify yeast species, and that this would somehow tell you which yeast species were related to one another. I'm going to show you that that idea really doesn't work out the way they thought it did, but it works out great for us. And so now we can use this trait matrix to look for positive and negative correlations. So we just did a bunch of permutation tests. This is a really busy figure because it's got all 48 traits. You see there are some positive correlations in blue, some negative correlations in red. Don't dwell too much on any of the traits. I'm just going to give you a couple punchlines here. One is that there are two major trait syndromes here. One is yeast pathogen syndrome, and that's that big cluster of red there, where things that tend to grow at 37 degrees tend to use fewer carbon sources. The other big one is a fermentation syndrome. And so this cluster here, that blue cluster, this is a bunch of fermentation traits. Those are highly correlated with one another, but they are negatively correlated with pentose phosphate, pentose metabolism, right? And so yeasts that are good fermenters tend not to consume pentoses, which is a real problem in bioenergy research because those are very abundant sugars in plant cell walls. If we now remove those two syndromes from the analysis and just look at carbon utilization, we can start to see some really interesting patterns, what I like to call metabolic allometry. So what's falling out here are actually the chemical structures. I want to stress here that there are no genes in this figure. No genes were harmed into making this figure. This is all ecological trait data from what they can eat that's coming out. So what happens when we actually add the genes? Well, we start to see that metabolic allometry makes a lot of sense. It starts to look like it's based on genetic pathways. So I'm going to highlight here galactose metabolism is right there. This is a sugar that requires GAL1, 7, and 10. We see that there's a strong association between galactose metabolism and the presence of these genes. I'm also going to zoom in on one example that really still amazes me to this day, and that is this sugar raffinose right here. It's actually a trisaccharide, 3 monosaccharides hooked together by linkages, and it looks like melibiose on one side and sucrose on the other side. And the gene that cleaves that bond is MEL1, and the gene that cleaves that bond is SUC2. And so across hundreds of millions of years of evolution, what we see are this structure coming out of the data where similar genes are being gained and lost. This is presence-absence data of the genes, and then the clustering is being done by the traits, and it drives this network. Okay, so a couple take-home points before we delve into the next mini stories here. Yeasts are extremely diverse. Hopefully you're convinced of that, as diverse as the whole animal kingdom. And these trait syndromes this concept is real. They're driven by carbon metabolism genes, and they're deeply embedded in the data. These carbon metabolism genes tend to come and go together in sweeps. So a few years ago, in 2018, we published the first tranche of data, and it is actually a little bit easier to orient with 300-ish species than it is more than 1,000. So we found that there are 12 major clades of yeasts. Those are color-coded. Those are now actually officially taxonomic orders. And we can now put some of the important yeasts onto a phylogeny. You can kind of see who's related to whom. There's the genus Saccharomyces, including the model yeast Saccharomyces cerevisiae, as well as a bunch of industrial hybrids. There's a whole genome duplication about 100 million years ago, so this gives you kind of a little bit of a scale. It actually involved this group and that group of yeasts in an allopolyploidization event. There's one other yeast you knew of before today. It's probably Candida albicans, one of the big baddies on the WHO opportunistic pathogen threat list, studied by Matt Anderson and others in the department. If you're a sour beer fan, Brettanomyces actually is involved in many sour beer fermentation. It actually evolved a very aggressive aerobic fermentation lifestyle independently of Saccharomyces cerevisiae. And then there's a few groups that contain yeasts of biotechnological interest, either because they produce lots of lipids or because they're able to ferment those plant sugars like xylose. Okay, so having a tree and a bunch of traits, one of the things we can do is march through time, backward in time from the tips through the various nodes, and ask whether we're seeing more gains and losses over the hundreds of millions of years. And it's not even close. There are more losses by a long shot. These are a list of traits in that data set that are statistically significant on their own. 1 to 16 is a blowout. If we look at traits that were not quite statistically significant, there is still a pattern there, 5 to 23. Again, maybe not as big of a blowout, but there's definitely a skew there because there's actually more samples there. It's a little bit more significant. And if you're laughing about why are you showing something as simple as a binomial test? Well, the reason this analysis is not as fancy as any of the others I've told you about or I'm going to tell you about is because I did this one. Okay, so the other thing we can do, in addition to just simple number keeping, is we can actually march back through time and look at the trend. And so here we're plotting in the center, the average number of carbon utilization traits that a budding yeast can use is the budding yeast common ancestor at the very beginning. And what we can see is that at least starting 200 million years ago, we see this strong downward trend. And so there's this general trend that budding yeasts are losing traits over time. Interestingly, though, the dots are all of the nodes in the data set, all the inferred ancestors. And what you see is that there's actually quite a distribution. There are always yeasts out there that can do more than other yeasts. And we'll come back to what those are in a bit. But first, we'll give you sort of the punchline of that prior study and some other earlier studies is that loss is really a major factor in yeast diversification. But that really raises a paradox. How can all of this loss generate the kind of diversity that we see, the different strategies that they have for what they're going to eat, when they're going to eat it, what they're going to do with it? How, and related to that, you'll hopefully see by the end, is what are the consequences of these interconnected carbon metabolism pathways, these that lead to these trait syndromes? Okay, so to resolve that paradox, we finished the project. We sequenced every known yeast genome, essentially. We also did quantitative growth curves on 24 conditions, 18 carbon sources and 6 nitrogen sources. We also created a new environmental ontology to describe in a hierarchical way, sort of like gene ontology, where all of the strains are coming from. So this is really a massive trait matrix. And I want to highlight a couple important facts here. Other than a handful of reference genomes, which we included for historical reasons, there's a 1-to-1-to-1 correspondence between everything that's in our freezer, everything that's been phenotyped, and everything that's been sequenced, right? So that's very important in making sure that we're not dealing with intraspecific variation or population variation within a species. I also want to mention that QR code you see there takes you to the newly revamped Y1000 Plus project website. You can download all the data. There are some curated highlight projects. So if you want to hear more about some of the stories I tell you or other ones, feel free to do that. Okay, so Here, and I think you can see why I started out with the 332, you can barely read any of them at this resolution, although some of that's a projector. But you can see we've highlighted some of our friends here. With 1,154 species, we can see that our sampling was pretty biased. Now the yellow group, the Serenales, that use a different genetic codon, they are massively expanded. And then in red, this order, the Dipodoscales is expanded, as is the Pichiales in orange. Saccharomycotales is down here, that dark blue, it has shrunk proportionately a lot. So now once we zoom out and have the full subphylum, we start to see where our bias is. There's some other interesting things to note here. Canada albicans has now kind of shifted down here. That was the pathogen I identified. We've spent a lot of talk about Canada auris, but it's actually a Serenale, but it's way up here on the phylogeny. And here's what used to be called Canada glabrata is actually very close to Saccharomyces cerevisiae. So pathogenesis is one of these things that has evolved many, many times. Okay, so there's a lot of data on this slide. So you're to be forgiven by saying, well, look, I'm trying to parse out where the isolation environment is, and I don't see a pattern. Well, that's because their yeast ecology is evolving very, very rapidly. There are lots of changes. And indeed, we can now look at our carbon utilization data. We can bin yeasts into generalists or specialists. And we use a statistical method to do this, but the end result is basically if you use a lot of carbon sources, you end up being a generalist, a little more than 10% of the data. Few carbon sources, you end up, again, a little more than 10% of the data, and about 75% of them end up in this standard bin. And if you look now at the ring that is yellow, blue, and purple, again, you can start to see some clusters of specialists or generalists there. But there's actually not that much of a phylogenetic pattern. And that is actually the point. That is where the power in the data set comes from. Because there's not much of a phylogenetic pattern, there are lots of transitions going on. I'm going to talk about a couple examples later in the talk where we're focused on individual traits instead of specialists and generalists. But again, the point is, all of these traits, they didn't evolve once or twice, they evolved over and over and over again. And that's where the power of the data set comes in. Okay, so one of the classic questions that we wanted to address in this framing of generalism versus specialism is whether there would be trade-offs in the data set, right? So this is the classic idea that the jack of all trades is the master of none. In other words, we might expect that specialists will grow better on carbon sources where they're specialized. We tested this idea in two ways with our data that I'm going to show you, but the punchline is that no, generalists grew better across the board. So first, you might have expected that specialists would go faster on the carbon sources where they are specialized, and because we have quantitative growth data, we can test that. No, they do not. The generalists are actually much more likely to be in this dark, fast category than the specialists are. And there's a trend there that's highly significant. This is even true if you only consider the carbon source where a specialist grows at its fastest rate, whether it's glucose or not. And indeed, there was no case where a specialist grew fastest on any of the 18 carbon sources that were tested. Second, you might have thought that there might be a trade-off between nitrogen and carbon metabolism. But instead, we see nitrogen breadth, and we only had 6 nitrogen sources, but there's still enough signal here that it's highly significant, there's no trade-off. If you're a nitrogen specialist, you're more likely to be a carbon specialist, and same thing for generalists. Okay, so I've told you at the beginning, and because this is a laboratory of genetics colloquium, nobody challenged me. I said, of course, it's going to be genes, right? It's always genes. Well, let's see what genes are driving this classification. So to do that, we applied a random forest machine learning algorithm, and this basically comes up with a decision tree to classify genes as generalists or specialists, and it worked quite well. We're getting about 90% accuracy and precision. For those of you that don't know how to interpret these, I'll just kind of run really through quickly. Basically, this is what you want to see if it's working well. If you say it's a specialist, it tends to be a specialist and not a generalist. So same thing, if you say it's a generalist, it tends to be a generalist and not a specialist. So the black diagonal, those are you're getting it right. The other white ones, those are where you're getting it wrong. This is called a confusion matrix, confusingly enough. On the right here is a receiving operating characteristic or area under the curve. And so what you want here is that red line diagonal is just random guessing. What you want is an upside down hockey stick. And so we're showing a few different iterations here, but basically we have an upside down hockey stick. And so what that means is that there is a lot of information. The random forest classifier is doing a reasonably good job putting yeasts in the bins of being generalists or specialists. Okay, so what is it using? That's the other nice thing you can get out of random forest is that you can get importance scores. And so I'm going to show you our top ranked genes, and it turns out that the top 4 genes that I'm going to show you also fall into metabolic pathways where we get other members of the pathway further down the list. And so the first one makes quite a bit of sense. It's complex 1 of the electron transport chain. This is highly variable in yeasts, and it's a major way that that energy budget gets managed by the cell. If you have complex 1, you get a lot more potential to generate ATP from a molecule of sugar. Beta-monosidase makes a fair bit of sense. This cleaves bonds and chains of mannose, which are really, really common in a fungal cell wall. And so there's a pretty good chance that most yeasts are consuming these at various points. Carnitine biosynthesis also comes up as very high on the list in terms of informing whether you're a generalist or specialist. And this makes sense because carnitine is actually used to kickstart respiration, which is highly variable in the data set, whether yeasts are able to use those respiratory carbon sources. And finally, the third one on the list is, and there are a number of genes in this pathway, is related to pentose and glucuronate interconversion. And so what this is telling us is that if you have a highly connected pentose phosphate pathway, you're much more likely to be a generalist. And so this is interesting because engineering flux through this pathway has turned out to be legendarily difficult, and that consumes a lot of my energy within the Great Lakes Bioenergy Research Center, figuring out how to get this pathway to run better. And it's interesting that we see it also coming through as really pivotal for explaining whether yeast is going to be a specialist or a generalist. So the next thing we wanted to ask is whether generalist and specialist evolved differently. So to assess that, we ranked carbon sources by how often they were used in generalists. And so that's what's going on left to right. Lactate is very rarely used, whereas glucose and sucrose are pretty much always used by generalists. We then used a Bayesian model to infer whether we're seeing more trait gain or trait loss in each case, specifically in generalists or across the tree as a whole. And so what we see here is in the rest of the tree, we see a lot of loss. That's that white there. And that's, I've already told you about that from an earlier data set and a different analysis, but it's good to see it come back here. But instead in generalists, we're seeing that there's not that widespread pattern of pervasive loss. And indeed, we're actually seeing some traits in black that are actually tending to be gained, such as xylose and cellobiose metabolism. So yes, there is a difference. Generalists are losing fewer traits and they're gaining more traits. So what impact does that pattern of loss and retention and reacquisition have on the genome as a whole? What has it had across these hundreds of millions of years? So to do something really simple, let's just count the number of protein coding genes in the genome, and you can see there's a distribution there. It varies a bit based on order. An interesting bit of trivia, the average yeast genome has 5,908, which I find remarkably close to Gaffo's Life with 6,000 Gene, the review article that accompanied the publication of the final version of the yeast genome. Now, it turns out that they were about 10% high on that, we know, but at least for a non-model yeast, it would have been pretty close to 6,000. And again, there's quite a bit of variation. The yeast with the fewest genomes is Stammarella lactis condensi, which has 3,775. And then there's one yeast here that's an outlier that's got over 20,000. Interestingly, both of those are in the same order, the Stipodascales order, and so there's a lot of variation in that order. Okay, so there's other things we can do with this other than just counting. We can ask if there's a correlation between the number of carbon sources and the number of genes, and there is, and it's statistically significant. So here we've got the number of coding sequences. It's not super significant, but there's a trend line there. Also, we were worried that this was being driven specifically by carbon metabolism genes, and the trend line still holds, even if you exclude those that are annotated for carbon metabolism. So this is a general trend here. More genes makes you able to use more carbon sources. Because of that trend line, we can even turn it into an equation that we would predict one carbon source takes 36 genes on average, 2 of which are clearly not annotated as ortho groups involved in carbon metabolism. Okay, so to summarize, not summer anymore, to summarize this part of the talk, I've showed you that loss is a major factor, but there are reacquisitions and gains occurring in the data set, so we can modify that take home a little bit. And I've shown you that generalists are actually growing more robustly pretty much across the board, and we've even implicated some specific pathways that are really predictive for whether you're a generalist or not, and that we think that's what's making them robust. I showed you that this difference between generalists and specialists is because generalists are losing fewer traits and even in some cases gaining more traits. And as I promised at the beginning, it's always the genes. Generalists have more genes because that's what is doing the action. Okay, so as I promised at the beginning, I now want to shift gears a little bit, and I'm going to focus on 3 examples where we can use the evolutionary dynamics seen in this data set to teach us about the genes behind 3 specific metabolic traits. And I want to remind you that the reason that this approach works is because those traits haven't evolved once or twice. They've evolved many times, sometimes dozens of times across the yeast phylogeny independently. So I'm going to start with a case that when we started working on it, we thought was pretty applied, but I hope by the end I convince you that it's actually yielded some fundamental insight. And so those traits here are the utilization of maltose and maltotriose. So for those of you that know our beer work, you know that malt extract or the wort that is used to make beer, the number 1 sugar is maltose, and it's 2 glucose molecules hooked together with an alpha-1,4 linkage. Maltotriose has the same linkage, it's just 1 more glucose molecule tacked on, maltotri-3, right? And so it's a bit bigger, a bit bulkier, harder to bring into the cell, and if you don't consume all the maltotriose, you're going to end up with kind of a sweet, cloying beer that most people wouldn't like, certainly not me. I want it all on the product side. Okay, so most beer is made with an interspecies hybrid of domesticated ale strain of Saccharomyces cerevisiae that hybridized with Saccharomyces eubayanus, which we and our collaborators discovered in the wild in 2011. So it turns out that in lager yeast, maltotriose utilization comes from the cerevisiae parent. And that trait you can see here is common in the top row there is common in cerevisiae strains, especially those that have been domesticated for bread or beer production. And you can see across there, it's actually quite rare in the genus Saccharomyces as a whole, including in Saccharomyces eubayanus, where it's absent in every strain that's been tested. So the model has been that maltose utilization is an ancestral trait that is shared widely across the genus. And it's these domesticated ale strains that have kind of become unusual, and they've acquired maltotriose transfer utilization so that they can make bread and beer for us. And then they simply brought it to lager strains when they hybridized with the wild versions eubayanus to get that cold fermented lager product. And because they're bringing those genes, they're dominant, and that's generally kind of been the main model. So it turns out you can adaptively evolve Saccharomyces eubayanus to use maltotriose. And so we did that so that we could get at the mechanisms by which maltotriose utilization could be adapted. And if you'll pardon my pun, there are 2 flavors of it that occurred. We're only going to focus on one of those flavors today. Actually, it's this one here, but this adaptively evolved lineage acquired the ability to use maltotriose. So how did it do that? Well, a previous graduate student in the lab, Emily Baker, about 5 years ago, showed that it did this by taking 2 genes that encoded specialized high specificity maltose transporters. And a gene conversion event actually took a patch from MALT3 and overwrote MALT4. And now, from these 2 high specificity maltose transporters that can't touch maltotriose, we end up getting a gene encoding a transporter that can consume maltotriose. Well, that's interesting because it already suggests, just Emily's results here that I've shown, and it turns out a group across the Atlantic a few months later got a very similar result in a different adaptive laboratory evolution experiment that they did. But we already can infer that there has to be some molecular epistasis here because the orange patch is doing something different when it's put in the context of the teal background than it did when it was in the context of the orange background. Remember, the orange transporter can't transport multitrios on its own, only when those sequences end up in the teal background. So after years of whittling it down and surviving a global pandemic, my PhD student, John Crandall, eventually determined that there were several specific mutations and indeed some higher order molecular, intramolecular interactions that are actually required to confirm multitrios utilization. And I'm not going to run through all of the data. He just defended his dissertation recently. So you should ask him about it. He knows the details better than me anyway. But if you look at these series of constructs, there's basically no case where you can make one or two changes and get all the way where you need to be to use multitrios. You have to have multiple interactions. They're interacting with one another epistatically in a lot of cases. And you can see here they're highlighted on the 3D protein structure. So because of the number of events, the number of changes required, this essentially created a situation where that gene conversion event that brought along 11 amino acid substitutions in a single event is essentially the only evolutionarily plausible trajectory, the only probabilistically plausible trajectory to get you there in a single step for selection to act on. Now, if you start to squint and do molecular docking simulations, as John has at the structure, and you start to squint at it, you can start to convince yourself how these changes have opened up the pocket in a way that we can now bind that bulkier multitrios, whereas before it was just able to bind maltose. So if that model is correct, if it's really about opening that channel up so that it can bind this bulkier sugar instead of being a high specificity maltose transporter, that really raises the question of how novel multitriose utilization is really. If it's about getting a open pocket rather than something that is very, very specific, such as for maltose, is that really hard to do? So it turns out, if we now look at genome sequences for more than 300 species from across the subphylum, again, this is hundreds of millions of years of evolution, you get this gene family has thousands and thousands of members, and we can build a tree with them. Some of them have well-known functions from certain systems, some of them don't. But what we see is that these high specificity maltose transporters that Saccharomyces eubayanus and Saccharomyces cerevisiae have are actually in a very narrow part of the phylogeny. You can actually barely see them here, right? And so they're embedded in these other transporters that are related to alpha glucoside transporters. And I want to stress that the vast majority of transporters on this tree have not been functionally characterized, right? So there's a little bit of extrapolation from the systems where it has been characterized. But the bottom line is the high specificity clade seems to be evolutionarily derived, and there's a generalist clade that looks like it may be able to transport things like multitrios. So to circle back to the original question, how novel is multitrios utilization anyway? Well, John went out and tested it in about 300 species, and it turns out it's not very novel at all. If we test across the subphylum, we see that many non-Saccharomyces yeasts can actually consume multitrios, and indeed a couple other alpha glucosides. Indeed, after phylogenetic correction, we see the number of these alpha glucoside transporters, the number of these generalist transporters, their copy number correlates with growth pretty well. Now, I want to pause here to acknowledge two reasons why the breadth of multitrios utilization has been missed before. First, it has been mainly of interest to people doing applied research in Saccharomyces related to beer production. And we're guilty of that ourselves. That's how we originally were thinking of it. Second, it's not routinely tested by taxonomists because it is very, very expensive. It's one of, I believe, 2 or 3 sugars that we have, bright labels on in the lab. Do not use without checking so that people don't make up liter batches of it. And even what you can get from multitrials is actually pretty impure. So the taxonomy data wouldn't be that clean anyway. And so this was the first time there was really a subphylum scale data set that could actually see this. And indeed, all the data I showed you up until this point, because of the cost and because of all of our biases that we brought to it, didn't contain multitrials. So we now have this emerging hypothesis that even though our specific AGT transporters that might be evolutionarily derived, have evolved to transport specific alpha glucosides in a specific context, they may have all evolved from generalist transporters, and there may still be generalist transporters out there. So in parallel, Kyle David, a postdoc in the Rokas lab, was interested in figuring out what might be the most important gene families for conferring carbon utilization traits. And so using a bunch of Y1000 plus data, as well as some other public data, he applied a novel phylogenetic regression approach, and he found that not all gene families are created equal. Some gene families, their copy number are actually controlling the evolution of multiple traits, and you'll see here in this distribution, there are those that can control 3 traits, 4, 5, 6, and the big outlier here is AGT, controlling a whopping 13 traits. And we can see here across the phylogeny, this is our 1100 plus species, and those heat maps are actually color coding the number of carbon sources that are being metabolized. And you can see that the AGT copy number is correlating with the number of carbon sources that are being utilized. So we call these keystone gene families because these gene families are having an outsized effect on the evolution. And we think that that's very similar and interesting to a concept from classic ecology, the keystone species. And so these are, just like keystone genes are influencing evolution of multiple traits, keystone species influence the evolution of entire ecosystems. So I'm going to do a dive here into this for those of you that don't know. This was actually my graduate advisor, Sean Carroll, but he wrote a book called The Serengeti Rules. To do a deep dive into sort of what a keystone species is, just for a moment, wildebeest are the lawnmowers of the Serengeti. And so during colonial times, colonialists brought cattle to what is now Tanzania, where the Serengeti is, and they introduced a virus called rinderpest. It decimated the wildebeest. 95% of them died. The ecosystem collapses. Fewer lions, fewer giraffes, and it's because of this lawn mowing function here. Fortunately, they eventually started vaccinating the cattle, and rinderpest goes away. The virus is gone. No longer detected in wildebeest. The wildebeest population comes back. There's less grass. They're doing their lawn mowing job, fewer fires. And you can see here from 1986 to 2011, there's a lot more tree cover. There's more lions. There's more giraffes. This is the effect of sort of a keystone species on an ecosystem. So the impact of these well-connected species really affects ecosystems in a way that we think is analogous to the outsized effect that we see for keystone gene families on metabolic evolution. And it turns out that, if you can remember back and haven't fallen asleep, I actually showed you AGT earlier when we analyzed presence absence data instead of copy number. But I labeled it, according to its best studies members, those MALT genes that are involved in maltose and multitriose metabolism, because, again, it's so important to beer fermentation on Saccharomyces. So that's how we thought about it in 2018. But now I think about it as a wildebeest. It's a gene that's been deeply embedded in the genome, guiding hundreds of millions of years of innovation across an entire subphylum. It just happens that one of those nice innovations is delicious beer. Okay, so one more take-home point. We have this concept of keystone gene families that are driving the evolution of many traits, probably responsible also for a lot of those trait syndromes we talked about earlier. So we can add that to the list of take-homes. And it looks like I have time to do the last two little deep dives here. So I'm going to shift to a fundamental carbon source. A carbon source has been studied to understand basic biology and fundamental biology for many years, and it's been my favorite since graduate school. So this is galactose metabolism, and that's because the yeast gal pathway has really just taught us so much about how eukaryotic gene regulation works. It graces our textbooks. It's also been used in all kinds of biotechnology, the yeast-2 hybrid system, the early method for detecting protein-protein interactions. The fly geneticists here use it in the gal4 UAS system, right? And so it's in addition to all the fundamental science it's given us, it's also given us some nice tools. So in more recent years, it's also taught us a lot about evolutionary patterns, especially loss and reacquisition, because the pathway has been lost dozens of times in evolution and reacquired a few times. So as the Y1000 plus data rolled in, I started to get bothered by the fact that the presence of the gal pathway didn't perfectly correlate with galactose metabolism. It was about 94%. Now you might think, well, for high throughput data, that's 94% is maybe okay, and it probably is okay. But some of the cases where there was a disconnect, we could actually go and carefully do experiments, and we were certain that the disconnect is real. They would either have galactose, the metabolism, but not the pathway, or they would have the pathway, but not be able to use galactose. So there's a gap in yeast genotype map. We're not able to predict a phenotype from a genotype that we should be able to predict, right? So what is that gap? Well, a gap in the genotype-phenotype map is an opportunity to discover new biology. And so here is the LOR pathway that's used in bacteria, humans, and textbooks, right? It's got those 3 genes we talked about earlier, GAL7, 1, and 10. But there must be another way that they're able to use it. So if we now apply the same machine learning approach that I showed you, GAL7, 1, and 10 come out. GAL1 is the most important. It's the first step in the pathway. That's maybe not that surprising. But we're still only able to get about 94% until we include some physiological traits. So if you notice, the physiological trait that is the most predictive is whether a yeast can utilize galactitol or not, which is just a sugar alcohol form of galactose. So that made us wonder if there could be some yeasts that are using an oxidoreductive pathway that goes through this galactitol intermediate. And it turns out that a pathway like that has been characterized in filamentous fungi. We think that the details are going to end up being a bit different in some cases, but it at least gives us some inspiration of what to look for. And indeed, that first step, a really clear prediction to develop a biochemical assay. And so Emily Ugalode, current PhD student, was actually able to detect that first step is actually occurring in these yeasts that lack the gal pathway, but are somehow using galactose. Emily has now done an RNA-seq experiment on these organisms that lack the canonical gal pathway, but can use galactose, and looked at what is upregulated specifically in galactose as opposed to other sugars. And what we can see is there are a bunch of genes here in red that are upregulated specifically in galactose. Remarkably, those genes fall on a gene cluster right next to a transcription factor homologous for gal4. Now, she's working on feverishly characterizing the pathway through gene targeting experiments now. So I would love to come back at some point, or better yet, have her come back and tell you about that characterization. But I don't know if you're a betting person or not, but that is the gene cluster. Okay, so I'm going to tell one more story, and this one will be about another sugar of both fundamental and applied interest, and that's xylose. This is the second most abundant sugar in plant cell walls, and so after glucose, and so it's absolutely critical. Metabolizing it and converting it to biofuels and bioproducts is absolutely crucial to figuring out how to use all that cellulosic biomass so that we can set up sustainable biorefineries and use all the energy in those feedstocks for valuable biofuels and bioproducts. The problem is, if you look at yeast genomes, and this is 300 plus species, you can't use gene content to predict whether they're going to use xylose or not. The vast majority of yeasts contain homologs for the 3 enzymes required, xyl1, 2, and 3. But I'll save you trouble looking for a pattern in this data set that there's not. You do about as well as random guessing. If you have a complete pathway, you have about a 52% chance of growth, which is not a bet that I would want to place. Famously, Saccharomyces cerevisiae, and we did some more rigorous phylogenetic analysis to clean this up, has genes that are actually homologous and indeed orthologous to species that are able to use xylose metabolism, but it still can't, cerevisiae can't use xylose. Even if you engineer cerevisiae, it doesn't do a great job of using xylose. Okay, so what does predict xylose metabolism across this phylogeny? Well, one hypothesis was that maybe the use of optimal codons would tell us. This is known to facilitate expression, especially in microbes. And so we looked across the xyl genes encoding the xyl enzymes. And sure enough, for qualitative growth, all 3 of the enzymes have more optimized codons if they are able to grow on xylose than if they are not able to grow on xylose. Only one of those actually survives phylogenetic correction, xyl3. Somebody wants to ask a question. I have some speculation about why that is. But it doesn't mean that codon optimization isn't mechanistically important. It probably is mechanistically important across all 3 genes. What it just means is that there aren't enough events, independent events, except in xyl3 to sort of get that statistical signal up there. Okay, so that's qualitative growth. We can now have something that correlates with qualitative growth. We can also look for quantitative growth because we have growth rate data. And it turns out that here, xyl2 is the only significant member after phylogenetic correction. So this is especially interesting because xyl2 is actually the likely bottleneck. And indeed, it has a reverse reaction. It's actually 10 times more thermodynamically favored than the forward catabolic reaction. And indeed, we see a lot of yeasts actually accumulate xylitol instead of going all the way and fully oxidizing that carbon source. And so we think that having an codon optimized xyl2 makes you more likely to be able to push through this thermodynamically unfavored pathway. Okay, so I've showed you we can use codon optimization in xyl2 for quantitative growth rates. Xyl3 has a role in qualitatively predicting growth or not. Probably the others too as well, although the phylogenetic signal doesn't hold up. What about xyl1? Well, xyl1 copy number plays a role. And so here what we're looking at is we've only colored the 3 genes, the boxes for the 3 genes, if they are multi-copied. And it's not super strong, but you can sort of squint and see the red dots where there is xylose growth tend to be present. And this is significant after phylogenetic correction, tend to be present where there are multiple copies of xyl1. And we actually think that this is likely really important for maintaining redox balance in the cell because this is another oxidoreductive pathway. Okay, and that gets me to our last take-home message, which is coming from the galactose and xylose story, right? These are cases where gaps in the genotype-phenotype map are really allowing us to reveal novel biology. And I think filling in these gaps is a major future direction. You know, I've shown you the things that we can explain from the data, and of course that is highly biased. There are lots of things that we can't explain. That's where the discoveries are going to come from, and that's where the excitement is going to be for the future. The other excitement is working with all of these great scientists who have actually done all the work. So I want to thank my whole lab, whether I had a chance to talk about your work today or not, as well as the entire Y1000 Plus project team. This has, of course, been a deep collaboration with Antonis Rokas at Vanderbilt University. Also, Clete Kurtzman and Meritz Grunewald on the taxonomy side, as well as a number of other yeast biodiversity researchers. And with that, it looks like I have a few minutes for questions. Carol.
Oh, that was really fun. I have a question about the generalist. I have 2 questions. One about the generalist specialist trade-off. It looks like the generalist is robust in a lot of ways. Is there a cost? What is the cost? And then my second question is, it's intriguing that the generalist is correlated with the AGT gene family expansion. Why? What's that doing?
Yeah, so I think, let me answer the first question first, because I think it's maybe easier if perhaps a little bit more speculated. But the AGT gene family, these are alpha glucoside transporters. And so to be an alpha glucoside, all that means is that you've got glucose hooked up to something with an alpha linkage. That universe of somethings is tremendous, right? Glucose is the most abundant monosaccharide on the planet. So anytime you hook it to something, if you use an alpha linkage instead of a beta linkage, you've got an alpha glucoside. And so what we think this is telling us is that probably a lot of these enzymes are pretty promiscuous and can bind to lots of different alpha glucosides. And indeed, the data from John, shows some examples of that. But probably there are cases like the maltose transporters that are evolving into specialists. And in the case of the maltose transporters, I didn't go through John's model in detail, but the idea is that they sort of became entrenched for a particular alpha glucoside, which they bind with high specificity because it's so important to Saccharomyces ecology. In order for them to get out of that rut, they have to make multiple changes at once to kind of bring them back to this sort of flexible alpha glucoside transporter that can bind maltotriose, maltose, and probably a bunch of other things. Okay, so to back up, why are generalists so good? Why aren't there trade-offs? Why are specialists so bad? I mean, we looked a lot. Nothing would have been easier, right, than to be able to say, hey, look, here's the trade-off. Everybody expected it, right? But we have analyzed the data in many different ways to try to find some evidence of a trade-off. So I think there's a couple possibilities. You could say, well, this is just a technical artifact, right? We have to, for convenience and to make the data comparable, we have to grow them all in laboratory medium at a particular temperature, at a particular pH. Maybe that's not right for some species. Maybe what makes them a specialist is that they grow at a higher pH in the gut of a particular species of rabbit at 42 degrees instead of 22 degrees. So that's possible. I don't know how we're ever going to answer that question. It may be a question for other people's lifetimes. But that's, we have to acknowledge that is a possibility. But I think another possibility is that, they're sort of as a consequence of them maybe getting into these very specialized niches, they're so specialized that there really maybe is only that kind of one thing that they can do well. And so the trade-off could be that they're just becoming really bad at everything else except for that one thing. And until we figure out what that one thing is that they're specialized in, the general trend in the data, if we just look at it objectively and say we're going to treat all carbon sources as equal and say, are they getting better at a carbon source or a suite of carbon sources, is that you'd be better off having these wildebeest gene families in the genome, being able to use lots of carbon sources, being well connected, having your metabolic pathways be well connected and having a lot of flexibility. You're going to be better off that way than having fewer genes and getting more canalized to particular paths. That's a long, very speculative answer, but Audrey.
Great talk. It's great stuff. So I was just thinking about launching off the xyl genes into a broader topic, which is using, so here you're correlating various features with phenotypes, but have you thought about entirely phenotype agnostic ways to discover new pathways? So maybe it's easier to launch off the xyl, that xyl2 case, where you see I'm guessing, you see a correlation between constraint on the codon usage and the phenotype. Can you look for other genes or features, whether it's copy number variation or constraint, that correlate with that phylogenetic pattern to find new genes that are involved in xylose utilization?
Yeah, I think that's a really interesting approach. and it's one that we are using, and have used in past stories, none of which I highlighted, decided to highlight today. But yeah, so we've looked at trait correlations and we looked at gene trait correlations, but I think what you're asking is gene correlations, right?
Yes, and
Yeah.
I'm thinking about extending beyond that, even like large language models apply to genomics where you don't know what the phenotype is, but you can, I bet there's a way to find signatures that will find new pathways.
Yeah, we've not used any large language models. I think that could be an interesting space to go in. We have done some of the gene correlations, and I think they're all mentioned. One thing that comes out of that is the Colloquium biosynthesis pathway for the secondary metabolite, right? This was really the first native budding yeast secondary metabolite gene cluster. Those genes are statistically associated with one another because you need two enzymes to make it, and then you need a transporter to bring it in, or you're making something that is locking up your own iron, and then you use also a transcription factor. So that you can find by doing that kind of gene correlation. There are other things we think related to mitochondrial function where there's some signal there, where we have not ready to publish or talk about those publicly yet, but I think there's definitely there definitely are cases where that's going to be informative. I think large language model will be interesting to try. I don't have anybody doing that, but it would be interesting to think about. Yeah.
So, if there are no trade, just to follow up on the specialist generalist issue, if there are no trade-offs involved, you would predict that there would be no specialists left. So obviously, either some kind of benefit to be a specialist, or alternatively, could the fact that you have specialists at all just reflect the fact that it's even that loss of function happens more readily than gain of function, and that the reason you have specialists is that you have loss of function until you can only just do one thing, as opposed to the adaptive value of being a specialist.
Yeah, no, I think you're absolutely right. I should have mentioned that as another model that I favor, and that kind of stems off of this figure here, right? Is that there's this kind of inexorable downward trajectory, but there's always things below the line. Those would be things that I would predict have sort of canalized themselves into a very narrow ecological niche, are likely to go extinct. If we come back 10, 50 million years, they're the ones that aren't there. And indeed, the ones that are on the trend line, the average for yeast, also might be getting down further. The ones that, I think we're going to see, the lineages that I think are more likely to see the future are the ones that are above the line, that are the generalists, that are able to do more things. They have more genes, they have more interconnected pathways. And so I think it's likely that, yeah, standard yeasts are evolving from generalist yeasts, specialists are evolving from standard yeasts. I'm gonna go to Brett, because Sam, you and I can talk afterwards.
I had exactly the same thought as Matt, so I'm glad to hear it. Makes sense. And what you just said about the probability of extinction makes me wonder if you have tried to just test that directly from the trees themselves. These phylogenetic models of extinction versus speciation, and you can estimate rates with some rather large confidence intervals, and lots of controversy there. But I think it does seem like what you're saying predicts that the extinction rate of specialist lineages should be higher.
Yeah, I agree completely. This is analysis that Kyle David in the Rokas lab is starting to delve into. We are well aware of the controversy, though, and so we are treading lightly. So for those who are not well aware of the controversy, is a little bit like a Heisenberg uncertainty principle here, that it's really hard to simultaneously measure extinction rates and speciation rates at the same time. You get these broad confidence intervals, and so.
But the difference is you have pretty compelling data that makes a strong prediction here. So you're not just going into it agnostically. Anyway. Yeah,
no, that's true. A few years, about 5 years ago, Jacob Steenwick produced a figure where we predicted mass extinction events that lined up with, the global mass extinction events. We haven't published that. Because I'm not sure we believe it, but, you know, but it's, but yeah, it's an area we're definitely thinking about and thinking about how we can get an answer that at least satisfies us enough to write a paper. But I think it's honestly, that's a question I probably wanted to answer more with this data set than but I'm not sure we believe the answer yet. Yeah.
I think you mentioned something about how the entire growth rate in the generalists for the specialists is probably due to the presence of specific pathways, and obviously the generalists would have more genes and probably more genetic pathways because it's more on carbon sources. Is it possible there's interplay between the pathways, like the fact that they have a specific pathway to utilize a certain carbon source might play a role in the growth rate?
It might play a Yeah, I think that is a really interesting question, and I don't think that it's one that we've answered yet. Somebody nod if we have and I've forgotten it, but and I would say so a lot of what I kind of jumped around a lot today, and we used the quantitative growth rate data a little bit to test this idea because we thought it was the cleanest test of something that a specialist should be fast on the thing that it's good at, right? That's intuitive. Most of what I showed you was presence and absence correlations of traits or genes and traits or gene trait, right? Most of it was presence and absence. There were also some copy number cases that were informative. I would say that despite how excited we were about generating the data, I think the part of the data set we've exploited the least and should be exploiting more in the future is the quantitative data. Some of the machine learning algorithms and other approaches are not as well suited to that kind of data, but I think it's a very rich data source that I definitely want to see mined to answer that kind of question. Yeah, Nicole.
So is this all there is? Is this all the yeast? Or are there more lurking out there?
Oh, I almost put in a taxogenomic section. But I cut it, and I don't even know that I have a. Do I have a slide? I don't. Okay. So, well, here. Since the lab was started, we've sent undergraduates into the field to find new yeast species. This is the wild yeast program. We're actually rebooting a third post-pandemic generation of it right now, and we have dozens of species. That are candidates for novel species. Some of them we have successfully described in the taxonomic literature. Taxonomists are so conservative, though, that they make it nearly impossible to do anything. But we're flying right through that. In fact, the thing I didn't tell you is that the Y1000 plus data set itself contains several dozen species that don't have names that are from the wild yeast program. And the taxogenomic part that I cut from the talk that I probably shouldn't have, is related to this here. So remember, taxonomists, when they describe a species, just because it's what they knew how to do in the pre-molecular era, they have species 785, and they score it for all these traits. But the traits are changing all the time. It's really not good for taxonomy. It's good for what we're using it for, I think. But now, at the exact same time that we discover a species, we have to score it for all those traits in order to describe it. You're not allowed to name it, until you do that. But we also are always doing it in the context of the genome. So, like to give one case, we named one species for Bucky many years ago before we had genomes. But when we described the yeast that we named for the Packers, which was literally isolated by an undergraduate who had actually worked in a sausage factory in Green Bay, you can't get much more Wisconsin than that. Part of the reason we did that, or named it for the Packers, was that. But the other reason is, it turns out that one of the diagnostic characteristics for its close relatives is whether it can consume lactose or not, of course, from the dairy industry. That's actually a pretty rare trait in yeasts. They can consume lots of other galactose-containing compounds, but lactose itself is pretty rare. And sure enough, because we had a genome sequence, we can look and we can find homologs for the beta-galactosidase and homologs for the lactose transporter in the genome, and that's included in the paper. And I think that's the future of taxonomy. That's the future of taxogenomics, is you get this type of richness at the same time you do the species description. It's not just a list of stuff they can and can't eat. It's also a list of the genes that are in the genome. Some of it's going to make sense. I mean, and this is, the other thing that kind of, drives me a little bit crazy about the taxonomic framework is that everyone we've done so far, there are traits that make sense, but there are also traits that don't make sense. So, galactose metabolism, that 94%, I think might actually be about as good as we're going to get for everything except maybe like glucose metabolism. And so I think the potential for the future of taxogenomics and for discovery is that each new branch is a chance to discover something new, especially if that branch has explored the same solution that other branches have, and if there's enough signal data to pull it out. So that's my hope for the future. I think that's how people should be doing taxonomy going forward. See what happens. Yeah, Jeremy.
Do you think there's a role for a large amount of horizontal gene transfer occurring among these lineages, given the great diversity in these traits across them?
I don't believe in horizontal gene transfer. All right, where did my cursor go? Yes, is the short answer. We'll do some backup slides here. This is some greatest hits material. Bunch of stuff gets lost in yeast metabolism. These are just some kind of, canonical examples. But after stuff gets lost, things get reacquired. 3 lineages of yeast we can see deeply lost their canonical gap pathway 100-ish million years ago. They all reacquired it by horizontal gene transfer. It all came from the Cereales order, which has a different mode of regulation than the Saccharomyces and Saccharomyces cerevisiae. We think that is a big part about why it's a donor. The Schizosaccharomyces pombe has also reacquired lactose metabolism from a Cereales donor. So that's one example. Some yeasts in this WS clade, this is Paulo Gonçalves' work, pretty recklessly discarded alcoholic fermentation, which makes them not very good yeasts in my mind, but there are still extant yeasts that can't make ethanol, they can't ferment. This turned out to be a pretty bad idea, and so many of the yeasts, and it's actually different parts of the WS clade did it in different ways, reacquired alcohol dehydrogenase genes from different groups of bacteria, and they also needed their pyruvate decarboxylase, so they actually took a native gene that they had that was optimized for another substrate and optimized it to work on pyruvate instead of phenylpyruvate. Really fascinating story, and I think there's more. The WS clade is so fascinating. There's so much cool stuff to come out of that. I told you, I didn't delve into much of this talk, but it has historically been thought that budding yeasts lack secondary metabolism. That's not true. There are some that natively can make some novel secondary metabolites. This WS clade actually grabbed an entire operon from a group related to Enterobacteriales, related to E. coli. They managed to figure out how to turn this polycistronic operon with 7 genes on and express them and polyadenylate them as eukaryotic genes. Another really fascinating story that I think is going to bear some fruit. And then that story is not about horizontal gene transfer, so I won't tell it. But yes, horizontal gene transfer happened.